Hello guys, welcome to this HL Bio lesson. Today we're going to be looking at proteins, which relates to section 2.4 of your book. First of all, the essential idea for 2.4 is that proteins have a wide range of functions in living things. So we'll be looking at some of these functions and talking about why they are able to have so many different functions. Our first understanding is this, that amino acids are linked together by condensation to form polypeptides. So again, that critical reaction there, condensation. We'll see some examples of this. First of all, let's review the generalized structure of an amino acid. And we can see it here. We have this central core of nitrogen and 2 carbon atoms. We have an amino group on the left-hand side, a carboxylic acid group or carboxyl group on the right-hand side, and we also have this R group. Now it's the R group, this yellow one here, where all of these amino acids differ. This generalized structure here is the same across all amino acids. Now, there are only 20 different amino acids that are actually used by cells and living organisms to synthesize proteins, generally. We'll talk about an exception in a second. Okay, so let's look at an example of a condensation reaction. This one here is between the amino acid alanine and the amino acid valine. We can see alanine here, and we can see valine here. So we can see the generalized structure there, and we can also see them differing in this R group here. You can see the R group of alanine is very simple, CH3, with a much more complicated R group in valine. Now the amino acids are linked by condensation reaction, and as always, water is removed in this reaction, and this forms a covalent bond. Now with carbohydrates, we saw the name of this bond is glycosidic bond. In proteins or between amino acids, it's called a peptide bond, but a relatively strong bond, sharing of electrons, peptide bond. The new amino acid chain is therefore called a polypeptide. Poly meaning many, so many peptide bonds, polypeptide. We can see here the covalent bond and the water has been removed. Okay, there are 20 different amino acids in polypeptides synthesized on ribosomes. Now, you might remember this from a previous course, ribosomes and protein synthesis. Here is a selection of 10 of the 20 amino acids. So again, we can see the generalized structure, always the same, and this R group in the box is the place where these amino acids differ. Now the 5 amino acids at the top are polar amino acids, and the 5 at the bottom are non-polar. This polarity, which essentially means that the molecules have a positive on a negative end or a more negative end than another, is important in determining the protein's final structure. We'll talk much more about this later. Note the R groups in the boxes, okay, the variable region, and check out cysteine, just interesting. You might remember that proteins, when we list the elements that they contain, it's C, H, O, N, but also S. And we can see this sulfur group here in cysteine. There's only a couple of amino acids which actually contain sulfur. Okay, the formation of polypeptides is controlled by DNA. Short sections of DNA coding for one particular polypeptide is called a gene. So a gene codes for one polypeptide, usually one characteristic. Cells can turn off or on the genes they need. This is called gene regulation. So not all genes are on at all times in all cells. You might remember this from the previous course. We can see here protein synthesis. Now, we won't spend a lot of time going into detail for protein synthesis now. We'll do a whole unit or section on that later. But as you can see, it's composed of two stages. The first involves transcription. So we can see that happening here. Now, this involves DNA being copied onto mRNA, and this occurs in the nucleus. So we can see here an mRNA copy of the gene is made in the nucleus. Now, the second stage is translation, and this is where we see the RNA being converted into an amino acid chain in the ribosome. So the translation actually happens in the cytoplasm on the ribosome, and this is where the amino acids are actually joined together into a polypeptide. All organisms' genes are also known as its genome or its genetic code for all of the possible polypeptides which can be synthesized in that organism. All polypeptides are made from the same monomer. So this is an amino acid, as you know. And here we have a selection. This is taken from your book, selected organisms and their approximate number of genes. So a couple of things to note, that first of all, virtually all organisms use the same 20 amino acids to construct their polypeptides, as we've said. The exception would be some cyanobacteria, for example. These are bacteria, special types of bacteria that live in hot springs, for example, in Yellowstone National Park. These actually contain a couple of extra enzymes to help them deal with that heat. So they actually have 22 amino acids involved. Another thing to note is that more genes do not necessarily mean more complex. So we can see here that rice has double the amounts of genes as human beings. So rice having 51,000, approximate 51,000 genes, whereas the humans have 20 to 25,000. Okay, you can see the second understanding there is very close to our essential idea that living organisms synthesize many different proteins with a wide range of functions. And the first application there is looking at a couple of these functions. So I definitely want you to have a few of these functions in mind moving forward. Although the first level of structure we've spoken about involves only 20 different amino acids, or usually, proteins have a wide variety of functions. To achieve this, proteins may form many three-dimensional structures. And there's actually 4 levels of structure we will look at: primary, secondary, tertiary, and quaternary. Okay, back to these specific examples. Here's table 2.8 from your book with some examples of proteins and their functions. So we can see here, I won't read them all to you, but if we just pick out some highlights here, we can see that rubisco is actually an enzyme which is important in photosynthesis. Insulin is a hormone very important to regulate blood sugar. Immunoglobulin is part of our immune system, and it's actually an antibody. Rhodopsin is a pigment which is found in the retina of the eye, so it's important for low light conditions. Collagen, which you may have seen in some sort of skin cream advertisements, is part of your connective tissue in the skin and other structures in the body, so this is a structural protein. And again, in terms of structure, spider silk that creates webs or a nest for spiders is also a fibrous protein. So you can see here a very wide array or range of functions. Okay, let's have a look at these different levels of structure. The primary protein structure is the sequence of amino acids within a protein as determined by genes and the genetic code. So this gets back to our idea of DNA, mRNA, and eventually a polypeptide. So this is the first level of protein structure. This is taken from your book. You can see a very simple diagram here with a free amine group on one end and a carboxylic acid group on the other. And we can see 1, 2, 3, 4, 5, 6 amino acids joined together. Now often proteins are many hundreds or even thousands of these amino acids. Here's another view. You can see it's actually a lot more complicated than that once you put in your R groups here. We can see the basic chain is very similar with this generalized structure, but then the R groups coming off the side here. And it's these R groups which often determine the next level of structure when we start to get other types of bonding involved. The primary structure determines the three-dimensional shape of the protein. Okay, now this basic amino acid sequence determines the three-dimensional conformation or shape of a protein. And we can see here, this relates to our secondary level of protein structure. So, secondary protein structure, the hydrogen bonds between the O and the H of different amino acids create repetitive shapes. And we have two general shapes you need to know. The first is the alpha helix, which creates a spiral staircase shape. So we can see here the alpha helix, this kind of spiral helical shape, and we can see the H bonds or hydrogen bonds forming between the O and the H of different amino acids, creating this shape. We also have the beta pleated sheet, a sheet with corrugated folds. So again, we see our hydrogen bonds here between the O and the H, creating this corrugated shape, known as a beta pleated sheet. So this is our secondary level of protein structure. Okay, we can also talk about a protein having tertiary and quaternary levels. So a protein may consist of a single protein or more than one protein linked together. And we'll see this is the case in quaternary structure. Tertiary protein structure is where a single polypeptide is in a globular shape. So we might think of the classic sort of enzyme structure, this rounded structure with a single polypeptide. Now notice the word there, single is important. In quaternary structure, protein structure, two or more polypeptides combine together to make a single functional protein. So the key difference here, tertiary has a single polypeptide folded into a particular shape. Quaternary protein structure means multiple, two or more polypeptides coming together to form a single functional protein. An example here would be hemoglobin. So we can see here the tertiary structure held together by a bunch of different bonds, including disulfide bonds you can see here. And when we get one of these polypeptide, which might be represented by the dark blue here, joined to another, the light blue, then this is considered a single functional protein, even though it's made up of two different polypeptides. And our example in the book is hemoglobin, a very important molecule in our blood, which basically helps to transport oxygen. So we can see here 1, 2, 3, 4 different polypeptides joined together, and we also have these heme groups in here which contain iron, and this is the place where oxygen actually attaches to the molecule. But this is considered one protein. Just a quick word about polypeptide and protein. Now these terms are often used interchangeably by teachers, myself included, but they are actually slightly different in nature. So let's have a look at the definitions. A protein is described as an organic substance consisting of covalently bonded amino acids and ready to carry out its function. That's a key phrase. For example, a protein might be an enzyme ready to catalyze a reaction. A polypeptide is a single amino acid chain with its own primary structure. The key difference, therefore, is in the molecule's preparedness for action. A protein is ready to do its job. A polypeptide might not be there yet. So, if this single polypeptide can perform its function, then it is a protein. However, if this polypeptide needs to be modified further or combined with other polypeptides, as is often the case, for example, hemoglobin, if it needs to do this to perform its function, then it is not considered to be a protein. And another point to note that hemoglobin, as mentioned, even though it's made of 4 different polypeptides, it is considered to be a single protein. So that's a good little look at the 2 definitions for polypeptide and protein. Okay, this understanding, every individual has a unique proteome. There's a new word for us. Each individual organism is genetically different from all other organisms. This is especially true if this organism reproduces sexually. As we have seen in 3.2 relating to natural selection, variation there is often determined by a sexually reproducing organism with meiosis, with crossing over, and independent assortment. A couple of terms. The genome, as we've seen, is the specific DNA sequence unique for one individual. So this is the sum of all of its genes. As we have seen, DNA codes for proteins. Generally speaking, one gene equals one protein. Now, the proteome is something different. It is the unique set of proteins, as coded for by an organism's genome, that they are capable of synthesizing. So again, the genome is all of the genes. The proteome is the sum total of all the proteins that could be coded by those genes. But please remember, gene regulation. This doesn't mean they are all being produced, but the organism can produce them if they turn on those particular genes. Nice little website. If you want to go to it, just do a Google search, the Human Protein Atlas. There's a few different things you can do there, but it's basically trying to track all of the different proteins in a human being, between 20 and 25,000 of them. So some nice little things to look at on that website. Okay, the final point we're going to talk about is this idea of denaturation of proteins by heat or deviation of pH from the optimum. Now, we've seen that proteins have a secondary, tertiary, and quaternary structure, which is held together by a variety of bonds. You don't really need to know these type of bonds, but some examples would be hydrogen, ionic, disulfide, and also these hydrophobic or hydrophilic interactions. Hydrogen are definitely ones you need to be able to talk about though. Now, these structures, these different levels of structures, may be altered by both high temperatures and changes in pH. Any alteration in a protein's ideal structure is called denaturation. And this denaturation may lead to a loss of protein function, for example, in enzymes. High temperatures basically mean more molecular movement. And this often means that the hydrogen bonds might break because the molecules are moving faster. Often, however, the stronger peptide bonds between amino acids remain intact. They're sharing electrons, these covalent bonds, they're generally much stronger than hydrogen bonds. Similarly, a high or low pH away from the protein's optimum pH or ideal pH can lead to shape changes. The extra hydrogen ions, which are acidic, or hydroxide ions, which are basic in solution, for example, in blood or cytoplasm, can prevent hydrogen bonds from forming and thus affect the shape and therefore the function of proteins. Okay, I've taken these graphs actually from, what would it be, 2.5, which is on enzymes. We won't spend a lot of time talking about enzyme theory before in the near future, but I want you to be aware of these two graphs just at the moment because they relate to what we're talking about. So we can see here, this is the rate of enzyme catalase reaction, so how quickly the enzyme is working, and here is increasing temperature. So as we can see, initially as the temperature increases, the movement of the molecules in a solution increases and the enzymes are more likely to react with the substrate, the things they're trying to break down or bond together. Once we get to a particular temperature here, this is working at its maximum rate. So this might be, in a human for example, 37 degrees. The enzyme's working well, it's functioning very quickly to its maximum. Now once we go beyond this point, once we increase the temperature up to 40, 45, 50 degrees, then the enzyme or the rate of reaction we can see starts to drop off. The enzyme starts to lose its shape. The enzyme is starting to denature. And once we get down here, all of the enzyme has totally denatured and our rate of enzyme reaction has stopped completely. So this is a very characteristic shape of the effect of temperature on enzyme reactions. Another graph over here shows the effect of pH. Now, we can see there's some similarity with this increasing sort of rate of reaction, but we can see on the other side there's also a fairly similar pattern here. So it doesn't drop off as steeply as the temperature does. Now what we're looking at here is we might have some specialized enzymes. For example, pepsin in the stomach has an optimum pH that is acidic, so it might be working around about a pH of 3. So as we approach its optimum temperature, the enzyme starts to work to its optimum or peak level, and then once we move beyond that, either side, we start to see the rate of reaction drop off. So this enzyme, pepsin for example, in the stomach would have an ideal pH of 3. We can see here also, this might be another enzyme just somewhere else in the body or in the digestive system, has an optimum temperature of 7. So what we see is the same pattern, not a steep drop off, but a fairly even drop off on both sides of this optimum temperature. And that's really all we need to talk about today. So we've really covered proteins, and what I thought I'd do is, this is taken straight from the book, but it's basically a summary of this section. So you can just pause it here when you need to and just check your understanding with these points. All you need is love? No, false. You need carbs, lipids, proteins, water, vitamins and minerals. Biochemistry, how cool is it? Hope you enjoyed, guys. I'll see you in class.